Oh, good morning, everybody. Welcome back as we start another chapter. We only have a few more chapters to go. In fact, we have 3 more chapters to go, counting this one. So we're getting very, very close to the end of the semester. But we've got some very important material still to cover. So in this chapter, we are going to continue looking at inheritance, and we're going to look at inheritance on the basis of chromosomal inheritance, focusing more on the inheritance of a whole chromosome rather than on the individual genes on that chromosome, although those individual genes do come into play as well. But it's more about the inheritance of a particular combination of chromosomes or number of chromosomes that are important here. So we have to cast our mind back a little ways in time, back to around 1901, when biologists finally started to recognize just how important Mendel's observations really were. And as they puzzled over what Mendel had been saying, remember he expressed everything in terms of statistics and probabilities, and people had to try and make sense of that math before they could make sense of what Mendel was actually proposing. But once they started to make sense of what Mendel was proposing in his papers, they realized, or they came to a realization, let's put it that way, that the only real way to explain Mendel's principles was that genes must be localized to chromosomes. Now, they weren't called genes at this point. That term hadn't been coined, and a large number of different terms would be used for Mendel's inheritance particles until finally someone proposed the term gene and it caught on. But the biologists of this period started to realize that the only way that you could explain the inheritance patterns that Mendel was proposing, or that Mendel had observed, was if the genes, these inheritance particles, were located on the chromosomes, considering that they were the only thing that passed from one cell to the next and from one generation of organisms to the next. So whatever was being inherited must be inherited via the chromosomes. But there was no real evidence for this. You couldn't say with absolute certainty that a specific gene was located on a specific chromosome. We can do much better than that today, however, with our modern technology. So this is a picture. It's a small section taken from a larger picture that's at the beginning of this chapter. And it shows a chromosome with some glowing spots on it. And what those 2 glowing spots that you can see here, and let me just highlight these with my pen. Let me see if I can make this stand out. Yeah, there we go. So these glowing spots represent the physical location or locus of a particular gene. And the way this is done is to make a synthetic DNA sequence of using a machine that creates synthetic DNA sequences. We've had these machines for about, I would say at least 30, nearly 40 years at this point. And they work very well. And the generations of machines that we have today are significantly superior to the machines that we first started out with. But you make this synthetic DNA sequence to base pair with part of the known sequence of the gene that you're trying to localize. You add a fluorescent tag to your synthetic DNA sequence, which we call a DNA probe, and you apply it to the chromosomes. And then you take a look under fluorescent light, under ultraviolet light, to see which chromosomes have picked up your probe. And you see the fluorescence. And you can see that it reinforces the fact that not only does the gene, not only is the gene localized to this chromosome, but because these are duplicated chromosomes, which is the easiest way to do this, because you can see the chromosomes quite easily, you can see that the 2 sister chromatids both contain the same gene. And if you look at the larger picture in the textbook, you're going to see that there is a second chromosome that has exactly the same glowing spots. And that tells you that that must be the homologous partner to this particular chromosome. So it actually highlights the 2 homologous partners. that we would expect to find. And so we can actually physically localize genes to specific chromosomes very, very easily. But back in 1901, all of this technology did not exist. And so it wasn't possible to do this. So the biologists were basically operating on the understanding that genes were actually localized to chromosomes. It was an assumption. And out of this would grow eventually what is known as the chromosomal theory of inheritance, which basically says that based on what Mendel had observed, the principle of segregation must be all about the separation of homologous pairs of chromosomes. And the principle of independent assortment must be all about multiple sets of homologous chromosomes being involved. So segregation is all about single pairs of homologous chromosomes. Independent assortment is about multiple pairs of chromosomes. And we know that from our discussion of meiosis a couple of chapters back, that this is in fact the case. And when we talked about Mendel's laws in the last presentation, I kept reinforcing the fact that these were the obvious physical manifestations of Mendel's laws, the behavior of the chromosomes during meiosis. And so having made that basic assumption that this must be what is going on, you can explain Mendel's laws by putting together this diagram. And you'll notice that it's talking about the dihybrid cross, because that includes both of Mendel's principles, because you see the law of segregation at work in the formation of the the separation of the individual pairs of homologous chromosomes that take place during meiosis 1 in metaphase 1. But you also see the law of independent assortment in metaphase 1, because you see the independent line up of the chromosomes. So you have the separation of the homologous pairs. Well, we should really say anaphase 1 rather than metaphase 1, correct? So the homologous pairs get separated during anaphase 1. The independent assortment takes place during metaphase 1. So we see in this, excuse me, in this diagram, the physical manifestation of Mendel's two laws. And that is how you can how you explain the ratios that Mendel proposed, the 3:1 phenotype ratio in the monohybrid cross, the 9:3 to 3:1 ratio in the dihybrid cross. The only explanation is that it involves these homologous pairs of chromosomes. But again, in 1901, all the way through to around 1913, 1914, there was no actual evidence that would confirm that chromosomes actually were the location of genes. But that would all change with the work of Thomas Hunt Morgan, who should rightly be considered to be the father of genetics research here in the United States. Mendel is the father of genetics research, period. I mean, he really started the discipline of genetics research. But Morgan is the person who here in the United States established the first genetics lab and started to produce some very interesting results working with a very humble little organism, the fruit fly, Drosophila melanogaster. Now, you have almost certainly seen Drosophila before. If you've ever left a banana out on the kitchen bench, you'll have noticed the tiny little flies that come buzzing around it. Those are the fruit flies. Now, when Morgan started working with fruit flies, or when he started doing genetics research, he didn't even start with fruit flies initially, but he realized very quickly after he'd found out about Mendel's work that to do really good genetics research, you had to have an organism that produced a lot of offspring and did so reasonably quickly. And while Mendel's peas were suitable for Mendel, being a monkey, remember, he had other duties, so he couldn't spend all his time working on his research. But for someone like Morgan, who was a research scientist based at a university, he needed to be able to produce results much more easily and much more quickly. And peas were simply not going to cut it. 3 months between generations was way too long. He needed something that produced offspring much more quickly and produced lots of them. And he was introduced to the fruit fly as a possible solution. Other people had started working with fruit flies, not specifically for genetics research, but one of his colleagues was an entomologist, someone who works with insects, and suggested that maybe fruit flies might be a potential organism to work with. The problem for Morgan was, number one, how do you go about growing fruit flies in the lab? And number two, because no one had actually worked with them before, it was not even clear that mutations would exist that you could work with, because you can't do genetics research without mutations. And so for Morgan and his students, the question became, how do we grow the fruit flies, number one, and how do we manipulate them? And then number two, how do we go about finding mutations? And Morgan and his students spent a lot of time trying to find mutations in fruit flies. The way we grow them is to actually grow them in little test tubes. So I can draw a diagram of this for you. So the test tubes have a flat bottom on them. You put a plug of agar plus sugar in the bottom. And you drop in a little piece of paper that has yeast on it. Because it turns out that the fruit flies don't actually eat the sugars. What they're eating are the yeast eating the sugars. So you have to inoculate the flask with yeast. Then you add your fruit flies. So a male and a female fruit fly, and you seal the top. And these days we use a sponge rubber plug to seal it, because it's not gas tight. We want the gases to go in and out. They need oxygen and they breathe out carbon dioxide like all animals. And the advantage for doing this is you can grow millions of fruit flies in a small space using these test tubes. And because they are flies and they quote unquote breed like flies, they produce a lot of offspring. You can have several dozen offspring in 2 weeks because their generation time is 2 weeks. And they're fairly easy to manipulate because you can knock them out with carbon dioxide. And so long as you don't flood them with too much carbon dioxide, you can keep them knocked out. And you can manipulate them under a dissecting microscope. They're large enough to see with a dissecting microscope or with a magnifying glass. And you can easily identify male from female. But that still did not provide Morgan with what he really needed, mutations. And he tried everything. He exposed the flies to X-rays. He tried different types of mutagens to try and encourage the formation of mutations without a whole lot of luck. And then one day, one of his students came to him and said, I've got one. I've got a mutation. So in order for you to understand what the mutation is and to understand a little bit about the flies, let's actually take a look at a male and a female fly, first of all. So here's a picture of a male and a female fly. And the male is on the right, the female is on the left. So let me put in the appropriate notation. You should all be familiar with these two symbols. So that's the symbol for male. This is the symbol for female. So wherever we have to talk about male and female, which we will have to do in this chapter, I'll often use those symbols just so that you realize which we're talking about. So this is male, this is female. So the male is always a little bit smaller than the female in terms of overall size. This is not unusual. In the animal kingdom, that is quite frequently the case. And you can see here the wild type features. So the red eye is the wild type color. Wild type, by the way, is the term that we use for the normal features, the normal phenotypes. So when we say wild type, we're talking about the normal features. So red eye is the normal eye color, and you can see that both the male and female have red eyes. They have these little hairs called halt hairs. They have antennae on their head. They have 2 of them. They have a pair of wings. The second pair of wings that many insects have do not develop in fruit flies, but they have the little nubbins or stubs that would develop into a second pair of wings should they evolve a second pair of wings, but they don't. They only have a single pair of wings. So this is true for all flies. Then the male is quite easy to distinguish from the female because he has this black region on his abdomen. So you've probably heard that insects have a head, a thorax, and an abdomen, those three sections. So there's the head. The thorax is the middle section that has the wings and the legs attached to it, and then you have the abdomen sticking out at the back. The male has this black coloration on the back of his abdomen, the last couple of sections of his abdomen. Female, as I said, is a little bit larger. She has all the basic features of a male, just a little bit bigger. She has black markings, but she doesn't have this distinctive black patch on the back of the abdomen that the male has. So even if you saw nothing else, you could easily pick a male from a female. Just look at the coloration of the back of the abdomen. Now, in this picture, the female is actually a little bit more obvious because this is an egg. So this female fly was knocked out, I assume she was knocked out at least, when she was in the process of laying an egg. And that's another way that you can actually pick the difference between the male and female, because the end of the female's abdomen has a little organ on there called an ovipositor that is used for laying eggs. That's what the term means, ovi, egg, positor, or egg layer. So that's another way of picking out the females from the males. So these are the wild types. And for the longest time, this is what Morgan and his students kept seeing as they were breeding their flies. They could not get mutations. And as much as they tried using all sorts of methods of manipulating the flies to get mutations, they simply could not get mutations. And it took them a while. But eventually, as I said, one of his students came to Morgan and said, I've got one, I've got a mutation. And here's the mutation. It's an eye color mutation. And this is not unusual, as you'll see. There are a number of mutations in fruit flies that affect the eyes. What that tells us is there are a number of genes that are associated with the eye color, because we know that there are multiple genes involved here. So this is the first mutation that Morgan and his students ever found, the white-eyed mutation. Now, it turned out that this mutant happened to be a male. And so Morgan knew that to capture this mutation, he would have to breed this male with a female. And the only females that he had at the time were red-eyed females. I mean, that was the normal feature that he was seeing. That's the wild type eye color. So not surprisingly, the vast majority of flies that he had were red-eyed. And Morgan assumed, as it would turn out correctly, but I'm sure it was a huge assumption on his part, that this female would actually be what Mendel would describe as being homozygous dominant. That the red eye was the most frequent feature that you would see, so that must be the dominant feature or the dominant phenotype, and that this female was likely homozygous dominant, as we would say today. And that was probably a fairly solid assumption to make, because if you think about it for a moment, if all you're seeing time after time after time is red eyes, you've got to believe that the red-eyed allele has got to be the most frequent allele in the population. And whenever you see some aberration, some deviation from red eyes, that's got to be the recessive trait. And therefore Morgan assumed that the male was going to be homozygous recessive. And he knew that this was the only way that he was going to be able to capture his white eye mutation. He needed to do that because he also knew, as we know, that if this male died without producing any offspring, his white-eyed mutation would die off with him. And so it was really important to Morgan that he capture this white-eye mutation by breeding the white-eyed male with a female, a red-eyed female, which he could assume was likely to be homozygous dominant. So this would be, in Morgan's mind at least, and I'm fairly confident that it was how he was thinking about it, this would be in Morgan's mind the equivalent of Mendel starting a monohybrid cross. The male is homozygous recessive, the female is homozygous dominant, and the expectation would be that the F1 offspring of this mating would all be red-eyed. And that's in fact what he found. Nothing unusual there. That's what would be his expectation based on what he knew about Mendel's observations. So now he has his F1 generation. He knows now how to recover the white-eyed mutation again, because he has his understanding of Mendel's work. He knows that he has to breed his F1 individuals and he'll recover his white-eyed mutation. So he sets up breeding pairs of F1 flies, male and female, in the flask, lets them breed, captures their offspring and takes a look. And sure enough, there are white-eyed mutants in amongst the red eyes. And the ratio turns out to be 3:1, exactly what he would expect for a Mendelian monohybrid cross. Nothing unusual here. Until he and his students took a closer look at the offspring. And then they got quite a surprise, because they realized that all the white-eyed members of the population, the F2 population, were males. There were no white-eyed females. There were just white-eyed males, which was something that you would not expect from Mendel's observations, because Mendel makes no point of talking about gender. He doesn't talk about male and female. Gender doesn't enter into the discussion at all. But here suddenly Morgan has something where gender apparently plays a role. Because remember, his original mutant was a white-eyed male. And while he didn't think very much of that at the time, having come through the F1 generation to the F2 generation and finding that only the males had white eyes, there were no white-eyed females, it now has the appearance of gender being important. That there's some role of gender in this inheritance pattern. But at this point, no one actually knew what was the basis for gender. So it was a little bit of a puzzle for Morgan at this point, exactly what was going on here. The answer would come from one of his former students, one of his former graduate students, in fact, one of the first female PhD geneticists in the United States. She was doing her own work at a small college in her free time. And she was working with some organisms called Japanese flower beetles, flower being F-L-O-U-R. These would be what we would call today weevils, a type of weevil. And she had been doing karyotypes of the males and the females. And she had discovered that the males had an odd pair of chromosomes that the females never had. The females all had true pairs of homologous chromosomes, and the males almost had true pairs of homologous chromosomes. But one pair of homologous chromosomes, or apparent homologous chromosomes, because they paired up during meiosis, were odd. They weren't exactly the same. There was a shape difference between the two chromosomes, so that even though they paired up as if they were homologous chromosomes, they were clearly quite distinct from each other. And only the males possessed this odd pair of chromosomes. And she decided that she would give letter designations to these two chromosomes. Now I'm going to see if I can find some space to draw a diagram. So the individual's name was Hetty Jakes, and she is the one that we give credit to for figuring out that gender is the product of chromosomes. Now, some people say sex, some people say gender. I was actually listening to a program just a couple of days ago on the radio that was talking all about this. And apparently there is some question as to whether these really should be called sex chromosomes, because there's a lot more going on here. The question of the biology of sex and the biology of gender, it's turning out to be far more complicated than just male and female. But I don't really have time to get into that discussion, but it's a very interesting discussion that one day, hopefully, I might be able to have with some students. But as far as this class is concerned, we don't have the time for that. But Hetty discovered that females had two chromosomes that were absolutely identical. So they were a true homologous pair of chromosomes. We can draw them like that. And she decided to call this the X chromosome, the X factor. It turns out that in the males, there's one copy of the X chromosome, and then there's this odd one, which she labeled the Y chromosome. And the Y chromosome is associated with male, the male gender. Only the males have the Y chromosome. And there's been some discussion as to why she named it the Y chromosome, and some people consider that it might be because of this odd shape. It looks a little bit like the top of the letter Y. The reality is a little bit more simple than that. She simply realized that the letter Y comes after the letter X in the alphabet, so XY. That's basically how she came up with the names for the two chromosomes. A second researcher, and I forget his name now, also arrived at a similar conclusion a little bit after Hetty discovered this. But Hetty passed on this information to Morgan, because she was one of Morgan's former graduate students. When Morgan received this information that in fact males and females in insects appear, or at least in some insects, appear to have chromosomes associated with gender, and specifically the males had a chromosome associated with maleness, that seemed to be the solution to his problem, because it explained the inheritance patterns. Now, you're going to have to work with me on this one. I've got a little bit of space in this slide, so I'm going to move this box up here so I can use the space down here to show you how Morgan eventually arrived at an understanding of what the inheritance pattern was here. So once Morgan had these white-eyed males, which you can see sitting right here, here's his white-eyed males, he started playing with different matings of his white-eyed males with other flies. He knew that mating his white-eyed males with what were probably homozygous dominant females would give him his F1 generation from which he could generate these F2. There's no problem with that. But he also knew that he was never going to get a white-eyed female. So he started playing with matings to see if he could produce a white-eyed female. And he hit upon a combination that actually gave him the white-eyed females. And it was one of his F1 females. So an F1 female crossed with a white male. And from that, he got white-eyed females. So he already had this information when Hetty passed on her observation that males had an odd pair of chromosomes that females didn't have. And only the males had that odd pair of chromosomes, and that must be the basis for maleness. It provided Morgan with his explanation. So how come? Well, if we use the designations that Hetty used for the chromosomes, the X and the Y, every male fly gets the Y chromosome. That should be a Y, not an X. Obviously. The females get an X in each. So why does that, how does this explain the patterns? Well, for a female to be white-eyed, she's got to be homozygous for it. Right? If we assume that the white-eyed trait is recessive, and there was no reason to think otherwise because it was the one that was missing in the F1 generation. Everyone was red-eyed in the F1 generation, and the white eye only turns up in the F2 generation. And it has a phenotype ratio of 3:1. So it's behaving as if it is a recessive trait. So there's no reason to think that it's not recessive. The only way the females could be homozygous for it is to have the white-eyed trait on the only chromosome that they get from both parents that is associated with their gender. And that has to be the X chromosome. In order for a female fly to have the white eyes, she must get an X with the white-eyed mutation on it from her father, and she must get an X with the white-eyed mutation on it from her mother. That's the only way she could be homozygous for it. It also explained why the white-eyed males existed and most frequently existed. That it was relatively easy to get a white-eyed male, a lot harder to get a white-eyed female. Because if the white-eyed mutation is in fact on the X chromosome, and the Y chromosome does not have an accompanying gene for eye color, then that explains why the males are white. They have the white-eyed mutation that they got from their mother, and that happens to be the only available allele, and that's why they have white eyes. And we now know that in fact that is the case. There is no matching eye color gene on the Y chromosome. So the only eye color gene in this case that the males can express is the white-eyed gene that they got from their mother, and is the eye color gene that they got from their mother. And if that happens to be the white eye mutation, then they're going to have white eyes. It is the fact that the inheritance of white eyes follows the inheritance of the sex chromosomes that is the solution to this problem, that explains why males predominantly have the white-eyed mutation, but you can get white-eyed females under specific conditions. And the specific condition is that the female must be a carrier, so she's got to be heterozygous. And the male must be white-eyed. Now, we do actually give a special name to the males who carry a single copy of the allele, because that's the only allele that they've got. We call them monozygous. So males in these situations are referred to as being monozygous. They can't be heterozygous, they can't be homozygous, because they are missing, they only have one copy of this gene, and therefore only one allele for the particular trait that they show. So it was the recognition that for a female to have white eyes, she must get the white-eyed mutation from both her mother and her father. But the only chromosome that's associated with gender that she gets from both her mother and her father is the X chromosome. Because obviously we still have to account for the gender basis of this inheritance. So it's got to have something to do with the gender chromosomes. That means it's got to have something to do with the X and the Y chromosome. But the only chromosome that the females get from both their parents is the X chromosome, which means that the white-eyed mutation, and therefore the gene for eye color that this white eye mutation represents, has to be on the X chromosome. And with this, Morgan is saying the location of one specific gene, this eye color gene, is now tied to one specific chromosome, the X chromosome. It is the only explanation for this specific behavior. So while previously people had to assume that Mendel's inheritance particles, what were becoming known as genes, had to be associated with chromosomes, there was no way using Mendel's observations to say that any one of those genes that he was referring to, that he worked with, was on any specific chromosome. But here, Morgan could now be very, very specific and say that the eye color gene that is represented by the white eye mutation must be located specifically on the X chromosome. It's the only way to explain the gender basis for this inheritance. A particular gene is located on a particular chromosome in this circumstance. And therefore, by extension, you could now say genes in general must be associated with chromosomes, must have specific loci on chromosomes, specific locations on chromosomes. And so for the first time, people could now not say we have to assume that genes are on chromosomes. People could now say we know that genes are on chromosomes and the specific genes are associated with specific chromosomes, all on the basis of this observation of Morgan. So, time for our first break, because we've got some more to talk about, because that's not the end of Morgan's work with fruit flies, as you'll see. So, here are our concept check questions for the first break. Remember, after the break, I'll give you the answers to these questions. Which of Mendel's laws describes the inheritance of alleles for a single character, and which law relates to the inheritance of alleles for two characters? And what's the physical basis for each of Mendel's laws? So this is basically asking us to remember what we were talking about in the previous chapter, because we have to be able to tie all of these inheritance patterns to the behaviors of chromosomes. So I'll see you on the other side of the break with the answers.